So this is going to be like a 2.1 review video. First I'm going to go over the functional groups and what molecules they are in. So from functional to molecules. If you want to review how to draw the molecules, I already posted a video on that. You can watch that video. And then after this I'm going to go over the study guide. So let's start with the functional groups. First we have the hydroxyl group. That's the OH because it has an oxygen and a hydrogen. It's called hydroxyl. These are found in carbohydrates. So like in ribose, you have your carbon backbone, and then you have the hydroxyl groups off of the carbons. So you need to remember which functional groups are in which molecules, that we can draw them properly. Hydroxyls are found in carbohydrates. The amine group, which is NH2. You have two H's off of the N, like that. This is the amine group. It's found in amino acids. Acids, because you have the N, H, and then it's connected to the C, connected to the other group, which is our carboxyl group. So there's your amino acid, and you have the amine group right here, which I'm boxing off in red, and then in pink is the carboxyl group. So we have the carboxyl group, which is R connected to carbon, connected to double bonded to O, and then OH. So you have the carb of the carboxyl, and then just like hydroxyl, you have the OH. These are found in your amino acids, but they are also found in lipids. You have the hydroxyls, carboxyls. In the lipids, look like you have the carboxyl here, and then you have CH2 and connected to CH3. So right there is your carboxyl. So it's important to know where the functional groups go for each of these molecules. That way you're able to draw them. It's easy if you follow the rules of just drawing the carbon backbone first, adding the functional groups, and then just filling in with any missing hydrogens like that. Okay, so if you want to further review how to draw the molecules, I already have that video up. Now we're going to go to the study guide. Anything that I don't answer in the study guide is not going to be on the quiz. However, for the test and for IB exams, you should review these questions and be able to answer these questions. But anything that I don't answer right now or that I skip over is not going to be on the quiz. So like I said in class, this one we will answer in topic 3 because it talks about DNA. Metabolic pathways are going to be chemical reactions that happen in a cell. So like the production of ATP or the creation of glucose during photosynthesis. It'll be one sort of chemical reaction, chemical pathway that's happening. And then for C, the term metabolism is all of the enzyme catalyzed reactions in a cell. So the difference is that like the metabolic pathway is one sort of pathway of chemical reactions that is happening, whereas metabolism is all of them. The web of metabolism, like we call it. So everything that is happening in that cell in terms of chemical reactions. For D and E, don't worry about them. They won't be on the quiz. Carbon is very important to this topic because, like we said, all organic molecules and compounds are going to be made from carbon. They have to be made from carbon, and they have to be a part of living things. So carbon is important because it is able to make 4 covalent bonds with different atoms, which makes it very versatile. Covalent bonds are also pretty strong. They're not the strongest, but they are strong. So carbon can form 4 bonds with up to 4 different atoms because it has 4 electrons on the outside that are ready to bond with other things, which could be other carbons, hydrogens, oxygens, whatever. But the important thing is you need to know that it makes 4, because when you draw them, you need to be able to count off and make sure that all of your carbons have the right number of bonds. So remember, carbon makes 4, oxygen makes 2, hydrogen makes 1, and nitrogen makes 3. These are the ones that you need to memorize in order to draw your molecules. Okay, so the carbon compounds that we were talking about, or the organic molecules that we were talking about: carbs, lipids, proteins, and nucleic acids. The key features of carbohydrates are that they are made in those ring structures. So they'll be pentagons or hexagons, like ribose and glucose, which are 2 of our examples. Anything really that ends in ose is going to be a carbohydrate or a sugar. Things like starch and fructose as well. The ratio of carbon, hydrogen, and oxygen in carbohydrates is that for every carbon, you have double the amount of hydrogen, but you'll have the same number of oxygens, usually, as carbon, or around the same. And carbohydrates, like we said earlier, have those hydroxyl groups. So in terms of the functional groups, they have the OHs on them. Lipids are fats. Their key features are that they are made up of long chains of carbon and hydrogen, CH2s, and that's where we have that little bracket with the N, meaning that there could be really long, big amounts of CH2s. So we use that N to be the variable for that. Again, they have the carboxyl groups, and that's the only functional group that they have. Examples, I would just say like fats or phospholipids could be another one. And they are made up of fatty acids, which you should know how to draw. Proteins are made up of amino acids. They are the only ones that have nitrogen other than nucleic acids, but we're not really going to talk about those. We'll talk about those more in topic 3. So they are the only ones that you know how to draw that have nitrogen in them. And that's because they have the amine group, which is NH2, and they have the carboxyl group. So the carboxyl group can be found in proteins and lipids, whereas the amine group is only found in proteins, and the hydroxyl group is only really found in carbohydrates. So like I said, nucleic acids we'll talk about more in topic 3. You should know that the examples are RNA and DNA, but that's about all you need to know for nucleic acids. So you should be able to fill out a chart like this to prepare for the exam and for your quiz. This is all really important information that you need to know. Okay, draw the simple structure of glucose and ribose, making sure that you have the correct number of carbons. So for ribose, I'll do it in pink. You start with the oxygen at the top, then you're going to make a pentagon like this with your carbons, but ribose is a pentose sugar, which means that it has 5 carbons. Right now we only have 4, so we put that CH2OH at the top right there on that left carbon. And then we're going to fill in with the functional groups that we know carbon needs to have in ribose, which is the hydroxyl groups, because hydroxyls go into the carbohydrates. This is a carbohydrate. You need to remember where they go, so they go at the bottom of these carbons and the top of this carbon. And then you just fill in with hydrogens to make sure that all of the carbons have 4 bonds. So 1, 2, 3. Put a hydrogen there. Hydrogens here to make sure these carbons have 4. And then a hydrogen there. So that's your ribose. And then I'll do glucose in blue. I will do alpha glucose just because it's not asking for a specific one, but you should know the difference between alpha and beta. So glucose, I start like that with the oxygen there so I don't forget. Then we have the carbon, 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 5. So glucose is a hexose sugar, so it should have 6 carbons. So just like we did with ribose, put the CH2OH at the top, and then you should know where the hydroxyl groups go from there. There's going to be 1 at the bottom of this carbon, 1 at the top of this one, 1 right here, and then since I'm drawing alpha, the OH would go at the bottom actually. Oops. Instead of the OH there. So remember how many hydroxyl groups you have and where they go. And then the rest is easy. Again, you just fill in with hydrogens to make sure that each carbon has 4 bonds. And then it's really easy. You just double check to make sure that they are all happy. Okay. So for this one, it's basically telling you the chemical formula of a fatty acid, and it's giving you that variable that we were talking about. So instead of writing it as this with the variable n and then the CH3, with this n you would just have the 6. So I would be happy if you drew it like this with your hydroxyl group there, or your carboxyl I mean, and then you have your CH2, but if you drew it like that with the 6 there and then CH3, that's okay. Or if you wanted to draw it all the way out, we have 1, 2, 3, 4, 5, 6, and then your CH3, and then just draw all the H's. That is also fine. You won't be asked to do something like this on the quiz, but you might be on the test. It's really just making sure that you know this generalized formula. Okay. Draw the generalized structure of an amino acid, labeling and annotating to show the different groups. So the general structure of the amino acid, you start off with your amine group, and that's going to be connected to your carbon, and then you have your carboxyl group, and then the H, and don't forget that variable. And then you would just annotate the different parts. So this is your amine group. And in another color, this is your carboxyl group. Okay. Alright, so identification of biochemicals. This is like what we did with the stations activity. So you might be shown a picture of a molecule and you will have to be able to identify it as one of the 4 that we were talking about or as one of the specific ones that you have drawn. For number 7, this one right here is tricky because you have like a long chain looking thing, but you also have this that kind of looks like a carbohydrate because it's a ring structure. But this is where the functional groups also come in handy. If you look, you have an amine group right here. Then you have the carboxyl group here. And all of this rest of it in the middle is your carbon variable, that R that we were talking about with the functional groups. So because it has an amine and a carboxyl, this molecule would be an amino acid. And it kind of tricked you with the C, the long carbon chain, because a lot of you looked at this and just assumed that it was a lipid because it has that long chain, but all of these C's are not connected to H's. So if it was like C, H, H all the way through, potentially, but it's not. It has an amino and a carboxyl group, so it is an amino acid. Which of these is glucose? It's going to be the one that is a hexose group. So a hexose sugar, so with 6 carbons, 1, 2, 3, 4, 5, 6, has the hydroxyl groups. So this one's glucose. The amino acid will have, again, the amine group and the carboxyl group. So it has to be this one. And then the one that looks like the fatty acid would have the carboxyl group and then those CH2 chains. So this would be the fatty acid. And then it doesn't ask you, but this one is ribose. And you can tell because it has 1, 2, 3, 4, 5 carbons. And it looks like the pentagon. The 2 that are most similar in structure are our glucose and our ribose, because they are both carbohydrates. So they have that ring structure, and they have the hydroxyls, so they look very similar to each other. Okay, so for anabolism and catabolism, both of these put together give you metabolism. Anabolism, like we talked about, is synthesis. It's creating bonds and creating molecules. Whereas catabolism, cats like to break things. It's breakdown. So you're breaking apart molecules to get monomers. The types of reaction. For catabolism, we have hydrolysis. And that's because hydro meaning water, lysis meaning to break, so water is breaking the bond. And then for anabolism, you have the condensation reaction. And we talk about these a little more down below in that diagram. Don't worry about substrates or enzymes involved. We'll talk about that more in 2.5, I believe. The products. So for anabolism, when you're creating something, when you're taking 2 monomers, or 3 or 4, and putting them together, your product will be a polymer. And then catabolism, when you're breaking apart those bonds of those big polymers, what you get are the monomers. So you get those tiny building blocks. And then for this one, you could have looked down here. For condensation, you are taking water out. So water is being produced. It is part of what will end up being the products of the reaction. Whereas for hydrolysis, it is being used, it is going in and breaking those bonds. Okay. Down here, again, don't really worry about it too much. This part is helpful because it shows like condensation and hydrolysis and like what the products are. For these, not too helpful. All of this, don't worry about it for the quiz. The test, you might be asked something about this, or on the IB exams you will be asked something about this. But you don't need to worry about it too much for the quiz. And down here I'll just draw what we had on the board for condensation and hydrolysis. So condensation, right, that was anabolism. And you're getting rid of water. So you would start off with 2 amino acids, like this. This is 1, plus the other, because chemical reactions kind of look like mathematical formulas sometimes, because you're adding things together to create that polymer. And then you have your little arrows. And then what happens is this OH and this hydrogen, they are going to come together to create water. So these two leave these molecules right here. They leave them to create water right here. And then what you're left with is these 2 amino acids, but they have that energy connected to them, so they still want to bond to each other. So what you end up having is a dipeptide being formed, because for this one, all you have left is this, double bonded to an O. And from this one, you lost that hydrogen, so you have N connected to a hydrogen, like that. So what happens is a bond forms between this carbon and this nitrogen to make sure that this carbon has 1, 2, 3, 4, and this nitrogen has 1, 2, 3, so that they're happy. So for the test and the IB exam, you will be asked to draw this out probably. For the quiz, you might be shown a picture of it, or you might be asked, like, what is a condensation reaction? And you would have to answer that. Okay. For hydrolysis, remember this is catabolism. And this is when you add water. Hydrolysis actually means water for hydro, and lysis means to break. So we're adding water to break a bond. So we would start with like our glucose. And this glucose is going to be bonded to another glucose, which gives us a disaccharide. Oh, it's so ugly. I'm so sorry. Let me fix that. It's still kind of ugly. And then we're adding water, remember? From up here, we're adding water. And then what we end up getting as a final product is that this water will insert itself, so all 3 of these elements the H, the other H, and the O add themselves to this. So the product is going to end up being two separate glucoses. We break it into the monomers. We end up having something that looks like this. So it looks like this. This O connects to one of these hydrogens right there. And then you have your other one right there. Ends up being like this. And this H and this O end up adding themselves to this molecule. So again, you're not going to be asked to draw this on any assessment, but you should be able to explain what is happening in a hydrolysis reaction, and you should be able to explain that it is catabolism that is happening, that they are breaking into monomers. So in this specific reaction, we started with a disaccharide. This was a disaccharide, and then we ended up with monosaccharides. Okay. So anything that I didn't cover in this video that is part of this study guide will not be on the quiz, so you will not be asked any questions about the synthesis of urea because we didn't go over that, but you will be asked about the different types of molecules, different types of organic molecules, different things related to metabolism. So make sure you review this video, you review the last video that I did, look back on your stop and jot, things like that. Okay?